Okay, thanks, Gerrit, again. We move on, and I think that we will have very little time for debate and questions from the audience in a more general setting, because we have to finish before 10. That means that the last speaker needs to finish before 10 o'clock, and the last speaker will definitely do that. Flühmann, Global Lead Non-Biological Complex Drugs at Vifor in Zurich. And he will talk about, clinical understanding and practical consideration for the use of nanomedicines. So finally we go to the clinic. Yes.
Thanks, Dan, for the trust in my speed. I hope you can follow if I speed up. As Dan said, we do a big leap forward towards the patient. I think the first time this morning where we talk about what happens in the clinics. Now I need to understand this thing here. No, the green. Okay, you have heard in the very first talk of Stefan, there were some differences observed with some of these nanomedicines in the clinic. This is an example in a hemodialysis patient. Hemodialysis patients are usually stabilized and they are HB with iron and ESA. It's nicely titrated, these two drugs, to keep their HB level in a target range. So what was observed when this IV iron was exchanged, switched, they dropped in their HB level, as you can see here on the gray arrow. You see then during these two periods, first period with the originator drug, 78 days in total, these patients were out of the target range, 99 days in period 2 with the copy drug. So they obviously could not reach the treatment target. How they had to correct was by adding more ESA, more iron. They had to give 34% more IV iron to these patients, and you have to be aware IV iron is not actively excreted, so it ends up somewhere in this patient. And 12.6% increase of ESA, which is a massive cost factor to the hospital. ESA is the erythropoietin. So, as we see, we observed clinical differences. There have been other studies, also more on the safety side. There were also other studies showing exactly the same thing as observed in these hemodialysis centers. And as Stefan nicely showed this morning, the authorities reacted to that. There was also a scientific discussion going on based on these findings. And that's where we're in, and I think we made huge step forwards in the science, in the regulatory science, to understand these things and address these things. What happened, these drugs have been launched about 10 years ago, a decade ago in Europe, without being aware of these facts. But then we had these studies. Now what we looked into, how do pharmacists evaluate these drugs? How do they react to this type of drugs? Are they knowledgeable about these things? So we asked them, iron sucrose similars, these follow-on products, do you consider them as just as effective as the originator product? And as you can see, 60% of them said they were neutral, 41% agreed they are the same. Same was for the safety aspect. They also said it's more or less the same. We don't see a difference. The authorities have to prove them as generics. They must be the same. Despite there was the clinical data out. Then we asked further, and that's for me more scary. We asked who takes the decision in the hospital what the patient gets. So you see, not even a quarter of the decision is taken by the physician or in agreement with the physician. Roughly half of it is taken by the pharmacist, And the last portion, the red portion, it's defined either in the formulary or in the protocols of the hospital. So, and we asked them, if you take the decision, pharmacists, do you inform the physician, the treating physician? And this was for me the most shocking result. 50% in Spain did not inform even the doctor that they switched the product. So doctors don't know with what they treat the patient. With drugs, we know they make a difference to the patient and the outcome of the treatment. We discussed this with several leading hospital pharmacists in Europe and came to the conclusion we need to do something in terms of education, giving them guidance how to better treat these patients. So we decided to make a roundtable with leading hospital pharmacists from Belgium, France, Germany, Spain, Switzerland, USA. Dan was part of this, not as a hospital pharmacist, but as an expert. So Netherlands was also represented there in a certain way. The goal was to reach a consensus on how to select nanosimilars in hospital formularies and how to evaluate substitution and interchange of these products. The approach we chose was we started with a guidance for biosimilars and looked what is relevant in this guidance for nanosimilars and what beyond that should also be looked at. The Liberal is a publication recommending a checklist of considerations for the formulary selection. This was the starting point. So you see here, that's from Griffiths et al. Adapted. These are the criteria they looked into for biosimilars. We checked all the criteria and came to the conclusion most of these or all of these criteria are also relevant for nano products. But then we thought about what is specific to nano products, and what is important is the physical characteristics, not only the chemistry, but also the physics. So if you could produce a nanoparticle, and I don't need to explain here in the group, there are two ways how you can go wrong by, they can aggregate or they can decay. And that's both not what you want to have in your product. So the manufacturing process is really describing these products, the physical characteristics. But what's also important, the storage and handling of these products. How are they stored? They have a shelf life of 3 years. In 3 years, a lot can happen to these very fragile components. So the physical stability is affected by this handling and storage. And these characteristics lead then to the size distribution, particle morphology changes, and so on, which we have heard in the first talk, influence PK clinical outcome. Based on that, we said there are additional criteria we have to evaluate before we exchange these products in a hospital. We have to look at the pharmaceutical quality and stress this point of this physical characteristic. Particle size, size distribution, particle surface characteristics, uncaptured pharmacological active moiety fraction, the labile iron, as we had the discussion before, and storage stability. These parameters must be measured along the time axis. On the efficacy and safety side, the pharmacokinetics is really, besides the uptake, the distribution of the nanoparticles is a relevant factor for nanoparticles. And a very important point is the stability for ready-to-use administration. If you dilute these products in saline solution for infusion, you need to make sure that after, if it reaches the patient, it's still stable. You still have the product you intend to give to the patient. And this data is missing for most of the products today, so you don't know what you have, and it might make huge differences. Based on that, we had the outcome, the selection criteria. based on the biosimilars criteria, we extended that with the necessary things for nanosimilars. The important focus points there were that the manufacturing defines the product, the physical stability and physicochemical reactivity of particles during shelf life and ready-to-use drug preparations, key things to look at. Then the particle interaction with the innate immune system, which influences the pharmacokinetic and pharmacodynamic profile of the drug, because many of the pharmacists, they just said it's not a protein, so immunology is not an issue. So we first had to educate them on this point. The whole list and the results of this workshop will be published in New York Academy of Science. It's in press. It should come out in the next weeks. That one is not yet online, but it should come out in the next weeks. With that, I give back 1 minute 47 to Dan, and thank you for your help.
Thank you, Beat, for your presentation. Indeed, very much the practical side. What are we doing with these nanomedicines, and in this case, then the iron sucrose? Are there any questions with respect to this presentation? Yes, please.
So I think I have a similar question for you. So what do you think are the most important physiochemical measures that are going to be to look at the biosimilars for the iron products? Because there's a lot listed, right? And
it's a very good question. Brings us back to the critical quality attributes, right? But what we for sure need to make sure is this physical form of the particle stays the same. And I think, and it's also something which fundamentally somehow changes, because it's not the content of an ingredient which is relevant, it's also the conformation. And I think that's something which was underestimated or not, people were not aware. And the far away you go from the, let's say, academia, the less they are aware what they're really handling there. So what we have seen that they, for example, if they see side reactions, they just dilute more, and we all know then these particles fall apart and you create even more side effects.
Okay, thank you. Any other questions for Beat? If not, then I think because of the time, we will close this session. The speakers will be around for discussion and interactions during the coffee break, at least some of them. But I would like to close with, first of all, thanking all the speakers for their presentations. Well, a little bit in contrast to my normal performance, I was very liberal in providing the time for the lectures. But then you have the disadvantage, of course, is that then the question and answer session sort of disappears. One last point, and that is a point that is related to all of the presentations, but in particular to the last one that Beat gave. What we really need is proof of how our NBCDs perform in practice. And so you need academic groups that run clinical experiments to prove similarity or equality or whatever you call it, sameness. And yesterday my esteemed colleague Gert Storm made the point that there is in the academic world, and that's where I go to Marissa, in the academic world, very little money to do that kind of work. And I think it's extremely important. But the innovator is not interested in it so much because it's already there, and generic companies, they get a product on the market anyway. So we really need some sort of mechanism, post-marketing surveillance system, where academics, independent, neutral groups can do this comparison. And there are examples of very good studies that have been done, but they are rare, very rare. And I really would like to also stimulate the idea that academic groups should work on this. And with that, thank you for the attention, thanks for the questions, thanks to speakers for their contributions, and it's time for coffee.